Good morning and welcome back. We have with us today Dr. Robert Looking. Let me introduce him first. Uh, Dr. Robert Looking obtained his PhD in natural science from University of Ulm, Germany in 1994. Since 2015 he works as a curator of lichens, fungi and bryophytes at Botanischer Garten of Free University at Berlin, Germany. Before that, he was there as a collection manager and adjunct curator in the Field Museum. And he has a wide interest for taxonomy, systematics, phylogeny, evolution, ecology, biogeography, and uses of lichens with emphasis on tropical taxa. And he's also interested in global fungal diversity and the formal classification of eDNA-based taxa and all. So he is a member of International Commission on Taxonomy of Fungi, which works on aspects of taxonomy and nomenclature of fungi. So this is what he has given, and we also printed in this e-abstract. But what is important to me and us, especially to the Indian lichen community, is that he was a highly helpful person. So whenever we have any doubt regarding identity of a taxa or any taxonomy complication, we simply write him a mail, and immediately the reply comes with a solution. So then whenever we write any manuscript, if it goes to him for reviewing, and certainly he would send it back with the positive comments. So that is what helping us a lot here, and that is helping us in improving our science communication and all. So such a helpful person. I especially wanted him to invite him for the this webinar, and luckily he agreed and he could spare some time with us, and we are all happy. I'm happy. So without wasting much time, I invite him for his talk entitled Lichens, Plant-like Miniature Ecosystem. Robert, please take over.
Thank you. Well, first of all, question, can you hear me?
Yes, very well.
Good. And also thanks for the nice introduction. I hope if you send me an email asking for identifications, I might not always be right, so I'm not sure how helpful I am, but thanks anyway. So I'm going to share my screen and see how this works. So can you see my presentation?
Yes, we can see it.
Good. So the idea of this talk is to give you a little overview over lichens, and I call this talk, I've subtitled it, Plant-like Miniature Ecosystems, because this is something that is emerging now as how we see lichens. So just to start out, I just want to give you a little overview of the diversity of lichens. I have selected here a couple of examples, some tropical temperate lichens, different growth forms, and you can see the diversity of colors, of shapes, of fruit body types. So lichens is a very diverse group of symbiotic organisms. Now, when people started out working with lichens, they were not aware of the fact that lichens are actually symbiotic organisms. They considered them an own group of organisms, and here we have a nice example of an early work by Fries, who was one of the pioneers in studying tropical lichens. And in his paper from 1825, he provided a very simple classification of lichens, and he already noted that some lichens are very similar to fungi and others are similar to algae. And this idea continued throughout several decades. Here we have another paper by Nylander in a Finnish lichenologist who worked most of his time in Paris, in France. Also an expert on tropical lichens. And he published a system where he also emphasized some similarities of some groups with algae and other groups with fungi. So lichens were seen as a group evolutionary intermediate between algae and fungi. Then, of course, in the late 1860s, well, I just wanted to show you this idea of lichens looking like other organisms. So there's lichens that look like liverworts, there's lichens that look like algae, there's lichens that look like macrofungi, and others that look like microfungi. So this is where this idea came from. And then in the late 1860s, of course, there was the discovery of the lichen symbiosis by Schwender and de Bary. In various works, they presented their ideas, and it became clear that actually these green cells or greenish cells that were visible in the algal thallus and were called gonidia at the time were actual algae or cyanobacteria. And so this idea of the lichen symbiosis emerged. Then there was, of course, a lot of different considerations how to define lichens, and one definition I like very much that was proposed by Trevor Goward in 1994 is that lichens are fungal farmers. So basically, they are fungi that discovered agriculture, and the plants, basically, or the photosynthetic organisms they cultivate are green algae, cyanobacteria, and some other types of algae. But more recently, the idea has emerged that lichens are more than dual or tripartite symbionts, but actually miniature ecosystems, because there's many other organisms involved found in the lichen thallus, and their roles are not quite clear, but it's obvious they're doing something there. So, for example, this paper by Spribilla et al. In 2016, they discovered basidiomycete yeasts in the lichen thallus, and very widely distributed, especially in the Parmeliaceae. And then there were quite a number of papers on the bacterial microbiome. So there's many different groups of bacteria that live in the lichen thallus, and they have different functions. Some are opportunistic, some seem to be more specific. And so this idea of defining lichens as miniature ecosystems was formalized in this recent paper by Hawksworth and Gruber in 2020. Now here's just an overview over some different definitions of lichens. So you see this goes back to quite early in lichen research. There's also some definitions from the 19th century, but this is a selection that I put together from different occasions, different papers. So, for example, in 1982, 1981, there was a poll by the International Association for Lichenology, what would be the best definition of a lichen, and this was then put forward by Ahmadyan in a paper in 1982. Then this definition by Hawksworth, lichen is a stable self-supporting association of microbiont and a photobiont, in which the microbiont is the ex-habitant, is a very popular one. It was a preferred definition for quite a long time. Then, of course, Goward lichens are fungi that they've taken up agriculture. Honegger's definition was more focusing on the lichen, the phenotype of the symbiosis as the result of these different components associating with each other. And then this most recent definition that a lichen is a self-sustaining ecosystem, including not only the principal symbionts, but other organisms. So what about the main bionts? So we have two groups of main bionts, it's the microbionts and the photobionts. The microbionts are only found in extant lichens in the Dicaria, in the true fungi, and about 99% are Ascomycota and 1% are Basidiomycota. The photobionts are systematically a bit more diverse. They are found in 2 different domains and 3 different kingdoms or supergroups. So there's green algae like Teboxia, Praseola, Tentapolia. There's cyanobacteria like Nostoc and Mysonema, and there's even some brown algae in marine lichen. What about global diversity and patterns and biomass, lichen biomass? So if we the most recent count is almost 20,000 lichenized species. As I said, most of these are Mycota. These represent about 1,000 genera, more than 100 families, and quite a large number of orders and different classes. And you can see here where most of the lichens actually found in one class of the Mycota, that's the Lecanoromycetes, which have about 75% of all known species. Now, when we look at the distribution of diversity of organisms on the planet, we often find this pattern here shown in the map of plant diversity, that most species or the highest diversity is found towards the tropics. Lichens were long believed that they don't follow this pattern. In fact, when you look at biomass distribution, you see that lichen-dominated ecosystems are mostly in cold temperate boreal regions or in high mountains. So this is an example of lichens in the tundra, and the reindeer is feeding on them. So it's mostly Cladonia species. And the tropics were believed to be not conspicuous in terms of lichen communities. And you can actually see there's a recent study published from Glacier National Park in Alaska, with a high number of species, I believe almost 900 species, and you see that the diversity is driven by landscape diversity. So in the different sections of the park, there's different communities that have a high number of unique species, and the number of shared species is much lower. And if you go at a higher regional level, so compare different regions in this area, you see a similar pattern. So there's lots of unique species confined to specific habitats, and the diversity is basically formed by habitat or landscape diversity. Now, if you look in the tropics, we have a different pattern. You have at a single site with homogeneous ecosystem, like a tropical rainforest, you can have more than 500 species within a hectare. On a single tree, there's currently a record set in a tree in Papua New Guinea. It's published by Uptrut a while ago, 173 species of lichens. And as you probably know, there's lichens growing on leaves in tropical regions, and there's up between 50 and 80 species have been found on single leaves. So you see there's a very high small-scale diversity. And you can also see that if you go in a tropical rainforest and look on tree trunks, there's a lot of different crustose lichens. And here's an example of 6 different species of Astrotelium. This is a very typical tropical crustose lichen genus in the Tripiteliaceae, just growing side by side. So what's happening when we look at a latitudinal gradient? So if you just look species by area, you see there's not a very distinct pattern. You might have a high number in boreal and temperate regions, and you might have high numbers in tropical regions. If you correct this by log area, because we know that there's a logarithmic relationship between species number and area, you see the pattern is even less pronounced. So there seems to be no gradient. But if you take into account habitat diversity, then you suddenly, this is quite a difference. So if you have a homogeneous habitat and you correct for log area, you see that in the tropics you have a much higher number of species at the small scale in single habitats. So that it seems the carrying capacity for lichens in the tropics is much greater, for different lichen species. Now I want to switch a little bit and look at the evolution of lichenization. Unfortunately, the fossil record for lichens is very sparse, and the further you go back in time, the more ambiguous is the interpretation of such fossils. So you can see this is a scheme that summarizes the main fossil finds that have been interpreted as lichens, and the ones in red are those that have been rejected. The blue ones are ambiguous, and the green ones are confirmed. And you see most of the green ones are younger than 500 million years ago. So everything else that is older and has been considered lichens is most likely not lichen. And to give an example, this is the famous Ediacarans from the Precambrian regions, about 635 million years ago. So these are macrofossils with different shapes, different morphologies, and these have been interpreted as lichens in part. But if you look at these, so these are some of these examples that have been thought to be possibly be lichens. If you look at their morphological features and you make a simple classistic analysis, you see that they actually cluster either with animals or with plants, but they do not cluster with fungi or lichens. And you can also see from the symmetry of these organisms that it does not fit into the concept of lichens. Here are some other examples of fossils interpreted as lichens and different interpretations. So, for example, the Sclerolichennomycetes, which is from Devonian, is currently accepted as lichen. Then we have the Spinfretia renticulata from about the same time, is ambiguous. And then we have an older fossil also from the Precambrian that has been rejected because there is no evidence that it actually is a lichen symbiosis. Then we have this interesting fossil where we have worked with our Chinese colleagues. It has been published recently. This is a fossil from the Jurassic, and you can see that it's interpreted as having these lacewing insects having a camouflage relationship with this supposedly fructicose lichen. And we've now been working on a paper that is coming out soon, where we reinterpreted the systematic position of this lichen. And so what we did, we compared it, we placed it into a molecular tree of the timeline of macrolichens, and you can see that most of the macrolichens are much younger than this fossil. So they cannot be placed in any of these families. And the only families that are as old or older than the fossil, they have a very distinct morphology and ecology. So the conclusion is that this is likely a new family. And this is the earliest well-documented lichen macrofossil, or macrolichen. Now, other than fossils, what you also can do is reconstruct molecular clocks with molecular data. And most of these point to an origin of extant lichen lineages at 300 million years ago. So some of these fossil lichens actually are older, which means they have to belong to lineages that are now extinct. And so this molecular clock approach gives you a lot of opportunities to look at evolution of lichen in terms of macro morphology and biology and macroevolutionary patterns. And here's a paper that we published recently looking at lichen growth forms evolution and associations with different algae. And what we found is some things are happening specifically in certain lineages, and in other characters there is some gains and losses all over the tree. So especially lichenization and delichenization seems to have happened several times in different groups of fungi. And here you can see some examples that we have been working with as well. This is a paper by Mino Tiaragaya and two papers, one on Stictidaceae, one on Artoniaceae, where the different lifestyles were reconstructed. So these are families where we have both lichenized and non-lichenized forms. And the question is always which of the two is more primitive and which is derived. And so actually very complex pictures arise here, and it depends really on on the particular situation, which is the case. Then also we have lichenization, as I said, in Basidium mycota. It's not many species and not many lineages, but a very interesting one is this Dictyonema lineage, which now includes 5 genera: Cephalostereum, Dictyonema, Acantholichen, Corella, and Cora. And you can see there's a very distinct progression in thallus morphology from a very primitive, undifferentiated quastose thallus in Cephalostereum, the filamentous thallus in Dictyonema, where the basidiomata become incorporated in the thallus. And then at the end, we have these highly complex foliose thalli in the genus Cora. So even if basidiolichens are not very diverse in number and there's not many lineages, there's also here, you can see there's models for the evolution of the lichen thallus. So I want to talk a little bit about lichen growth forms. As I showed you, there's many different morphologies, but there's a few principal growth forms. So one, probably the best known one is foliose lichens. And we believe from studies that they produce large biomass, so usually have to grow. They need like physiological optimal conditions to be able to produce these conspicuous thalli. And these lichens are highly competitive, so they can overgrow other organisms, and they thrive in a highly competitive environment, but only if conditions are right for these. Then another group, morphological group, are crustose lichens. So these can cover large areas even with low biomass, because they're basically completely adnate to the substrate and with very thin crustose thalli. And so they can tolerate certain kinds of stress, for example, water stress. And even so grow to relatively large or light stress or low light conditions to relatively large area. And then we have fruticose lichens. They also can develop high biomass. They escape competition because they grow into the open space. And usually these thalli are able to condensate water from atmospheric humidity. So these are more common in situations where most of the precipitation is happening in terms of clouds or fog, for example, coastal deserts. And there's also an interesting group of so-called dimorphic lichens. So usually they form a basal thallus and they form erect structures that, for example, in Cladonia are called podacia, that may or may not produce fruiting bodies. And so these usually grow on uneven substrata, on soil, or very rough structured bark. And usually these elongate structures or the erect structures help to raise the spore-bearing structures above the surface. Now, when we think of lichens in ecosystems, there's many roles that lichens have. For example, as epiphytes, they participate in the water cycle. They buffer precipitation and release it into the ecosystem. But of course, one of the most important functions is the nitrogen fixation by lichens that associate with cyanobacteria. And in ecosystems where this high biomass of this kind of lichens, the nitrogen input can be quite substantial. And as you know, only bacteria, cyanobacteria, and some other groups of bacteria are able to fix atmospheric nitrogen. Then there's also interactions with animals. So we know that many birds use lichens in nests. For example, hummingbirds are known for this. And it's believed that this because these lichens have antibiotic properties, the substances they produce, especially the cortical substances. There's lichens involved in camouflage. So here's 2 examples of a frog and grasshopper that have skin or wing patterns that mimic lichen communities. And interestingly, there's many lichens that grow on man-made substrata. So we know from concrete in urban areas, but also here, if you leave an artificial substrate long enough in the wild, you see it becomes overgrown by quite a diverse lichen community here in this case. And I especially like this example. This is from a photo by Gail McMartin from Great Britain, an old truck that has been left in the forest. And you can see the diversity of lichens growing on this vehicle. Now, when we look at lichen habitats, most diversity is found in well-conserved habitats. So lichens, even if they evolved to be able to exist in very extreme environments, they're very sensitive to environmental changes. And unfortunately, many of these conserved habitats nowadays, they look like this. And you can imagine that not only for lichens, but for many other organisms, this is catastrophic because the diversity of microhabitats, niches, completely disappears when we have such, in this case, tree monocultures. And there have been quite a number of studies on the effects on lichen communities. And here's one that has been done in Colombia, where we can see that if you compare a natural forest, mostly dominated by quercus trees, with an eucalyptus plantation, you see that the diversity, overall the diversity of lichen decreases. And this is because each of these eucalyptus trees supports quite a number of species, but each tree has the same species. So you have like a homogenization, and there's no diversity produced by which you would have in natural forests, where you have different tree species and even species, trees of the same species like quercus. Every tree has a different lichen community. So this effect is pretty well documented when you have monospecific tree plantations. Then, of course, there have been many studies, lichens in air quality, starting in the late 60s and then throughout the 70s and 80s. And these have, in many parts of the world, they have, especially Europe and North America, have led to quite an improvement in air quality, because based on these studies, there were measures taken, like filters and catalysts for cars. But we can also use lichens for these to assess ecosystem health. So this is an example of a study we did with assessing different lichen families and their performance or diversity in different under different conditions. And we found, for example, these Telotremoid graphidaceae are very good indicators of a healthy forest ecosystems. And also the Lobaria peltigeraceae. So these are two groups that are very good indicators. And we can measure what is called the index of ecological continuity. And here you can see there's a correlation between lichen diversity of these groups and the disturbance condition, and you see there's a very strong correlation actually. So the more diverse these groups, the less is the disturbance in a particular area. And here's a similar case where we looked at 4 species of Sticta, and we found that these have actually very different sensitivity to environmental disturbance. And this is done in New Zealand, and you can see there's 2 species that are a little more tolerant, and there's 2 species that are highly sensitive. And we can use this to make a very simple protocol for monitoring different sites and assess their conservation status. And especially in these island ecosystems, this is important because these are usually highly disturbed and highly fragile ecosystems. And an example also here, we can use lichens to monitor climate change. So this is a study that was done in Brazil, looking at lichen communities along the Atlantic rainforest. And you can see that in this graph down here, that looks at different environmental parameters, climate parameters, that actually they, along the latitudinal gradient, they change in a different way. And if you model this for species, lichen species in this area, many of these will actually shift their distribution to the south, or they will have a reduced range. And overall, the effect is that these tropical communities will move, for example, we predicted within the next 50 years, about 50 kilometers to the south. So there's an extension of tropical communities and a reduction of these temperate lichen communities. Of course, also interesting when we look at lichens is their secondary metabolite chemistry. And I mean, there's been thousands of studies literally on this subject, and I just want to touch this very briefly. One thing that's interesting that we looked at, for example, lichens in the lichen thallus compared to the lichen microbiome in culture often produce different substances, and there have been many other studies showing this, also especially in Graphidaceae, but also in other groups. Overall, there have been a lot of basic studies doing bioassays and this kind of approaches, but there's very little going in terms of really clinical applications. So most of lichen substances are actually used in traditional medicine or in homeopathic medicine. And so overall, for example, this project in Costa Rica that has been started in the 90s between Merck and the National InBio Institute, there was really little outcome in terms of the number of natural products that may be able to be found in plants, lichens, and fungi. So this is actually it's on one hand it's a promising subject. On the other hand, the results are compared to the effort that has been done not very substantial. And so I want to finish with my talk with another aspect of lichen chemistry, and that is the UV fluorescence. And that's usually something that you can use to hook people on lichens. So when you go especially in a tropical forest at night and you have a UV lamp, this is what happens. So you look at this tree trunk and you see it's a lot of different white, mostly sterile lichen thalli that don't look like anything too exciting. And then you shine UV light on them and you get this. So these have different fluorescent compounds. And of course, these can be used for taxonomy, but they're also interesting in terms of their chemistry. So what are the substances behind these fluorescent reactions? And I'll just give you some examples here. This is a diorygma. This is an unknown quastose lichen, and I have a perenniula, and here's Berthosaria. And so we dubbed this the enchanted jungle. It's very nice when you have a UV lamp and you can take this at night into the forest. So with this, I want to finish my talk and want to thank, and if there's questions, I'm happy to answer them.
Okay, very nice talk by Dr. Robert. So now it's open for discussion. If there is any questions, you can ask.
We have ample time.
Yeah, we have ample of time. So there are a few questions in the chat box here. Of course, Sebastian Selva is asking how we can identify fungal and algal partner. It's a very basic question. If you would like to answer, Dr. Robert.
Well, that's a good question. So the identification of lichens is actually first of all, we have to take into account that the name that we give to lichens, the scientific name, is the name of the lichen microbiome. So if I call something Cladonia rangiferina, and it's the fungus that forms the lichen that we recognize, but it's only the fungus. So the photobiont has a different name. And for example, many lichens associate with photobionts of the genus Triptychia. It's a green alga is a common photobiont genus. And so when we identify the lichen, we identify the lichen microbiont. And if we want to identify the photobiont, then of course we have to look at characters of the photobiont. And oftentimes this is much more difficult because the photobiont is microscopic, and to be really sure, if you don't know exactly what you have to culture it or you have to sequence it. But in many groups, at least we can identify the photobiont genus. Not sure if that answers the questions.
Yeah. There is one more question by Arun Christy Sebastian. What are the different aspects used for lichen biomonitoring?
Well, lichen biomonitoring, there's two approaches. One is active and one is passive. So the passive monitoring is basically mapping the lichens present in a particular situations, and then you have to compare this to a control. So, for example, when we do urban lichen mapping to see the effects of air quality, in theory, we need to know what lichens would be present if there was no pollution. Often this is difficult because, an urban area has been developed for so long that we don't know what lichens were originally present. So, but we can use areas like parks and other areas that have an extended range of not natural, but green vegetation, and use this as a control, and compare this then on lichens growing on trees along roads, and compare this, for example, with traffic patterns. And oftentimes it's also good if you have many cities now have stations that measure the major pollutants, and you can correlate that with lichen communities present at these stations. And so this enables you to map huge areas, for example, of a city with very little effort, and make maps of different contamination zones. Then the other approach would be active monitoring, where you actually use, for example, a particular lichen species. You collect it, for example, in other areas, nearby forest, and you have to use a species that is relatively common, of course. And then you can basically transplant the species to different parts of a city, and then for a prolonged time, a couple of months, a year or so, and see what the effects are on the lichens. If the lichens survive, if they're damaged, what the degree of damage is. And of course, all these aspects we can also apply to other situations, not only air quality, but ecosystem health, climate change. So the basic idea is always the same. There's active and passive monitoring. And it's very important, of course, is the taxonomy. If you don't know your lichens, then it's not really helpful if you do lichen mapping, because you cannot really interpret the data. So the taxonomy beneath all these efforts has to be sound.
Yeah, okay. Next, we have a question that...
Sanjay, I want a question to ask. Looking,
go ahead.
I'm Upreti from NBRI, Lucknow. I think we all know that the present number of species of lichens throughout the world is around 20,000. What is your estimate? Means what will happen? Means how far we will go, or what will be the extent of these lichen species in the world? What do you think?
That's a very good question. I mean, if you look at fungi in general, there have been predictions that are more than 10 times the actual number. So right now there's 150,000 species of fungi known, including lichens, and the predictions is at least 1.5 million. So we could think of we can transfer this one-to-one to lichens, but I don't think that's the case, because lichens is, among all fungi, lichens is, in my opinion, the best known, the best studied group. Because in contrary to other fungi, lichens have an open vegetative thallus, a permanent thallus that can be studied, has a lot of characters, while fungi grow with hidden mycelia, and you only see the fruiting, the spore-bearing structures. So this is why lichens have been studied for a long time in much detail, and while we can expect there's many undiscovered species, I don't think it's 10 times as many. So my estimate would be we have probably 2/3 of the species recognized, and the final number will be somewhere around 30,000. But this is really hard to predict, because now we see when we start studying groups of lichens that we thought we know, and we do molecular studies, we see that there's a lot of hidden diversity in these groups, and especially in lichens that have been thought to be widespread. So if you have a species that is believed to be cosmopolitan, some species really are, and in some cases really don't know why and how this happened. But in many of these cases, there's regionally differentiated species that often are not recognizable only by the morphology. So you have to do molecular studies. And in some of these groups, actually the number of species explodes. But I think this is more an exception. So overall, my guess would be there's maybe another 10,000 species to discover, which is a lot, but it's not as much as other groups of fungi.
Thank you.
Actually, what happens is we in our presentations update the number of lichens for India frequently. Like sometimes back we used to say 2,900 species. Then now we say, now we say that it's almost reaching 3,000 species. But when it comes to world number of lichens, we since several years we're telling 20,000 only. So in a previous one of my presentations, somebody asked, see, in Indian case, the number of lichens list is increasing, but the world it remains the same, 20,000. Why it is so? So it's a difficult question to answer. So.
Well, the reason is, of course, the world number also increases. So the last number that we had in 2017 was, close to 20,000. If I now would make a new number, I'm probably more closer to 21,000. But for example, if you look at individual regions and countries, of course, the number increase in part because you find new species, but also in part you find species that have already been known from somewhere else, and it's new records for the country. So they don't increase the global diversity, but they increase the diversity for the country. But I mean, India is a good example why I would say 3,000 is still much underestimated. I mean, based on the size and diversity of ecosystems you have and the evolutionary history, I would expect you should have distinctly over 5,000 species. So lots of work to be done.
Yes, really.
Actually, Sanjeeva, just one point. Like Dr. Robert, I have come across with in a national park, okay, and there the lot of lichens are growing, which is hanging like it is a fruticose lichens. So they are growing. So the persons who are staying in that area, they highlight that the particularly the monkeys and the elephant and rhinos, sometimes they come and I don't know, they eat or they simply see that. But I have a strong opinion that maybe a lot of birds and a lot of monkeys, they are sometimes used as a food. So have you worked on this animal and lichen interaction or dependency? Mm-hmm. If so,
well,
then yes.
Yeah, I mean, it's known that interactions between animals and lichens are very diverse, and animals using lichens as food is also well known in some cases. I mean, reindeer is the best known case, but there's also macrovertebrates eating lichens. Many herbivores, at least in part, have lichens as part of their diet. Of course, it's usually obvious in ecosystems where certain lichen morphotypes are abundant. So these cladonias in tundra, where they really cover large areas, of course, this is an important alternative food for macro herbivores. In tropical areas, I'm sure this also happens, but it's not well studied. But we know, for example, on another scale, if you look at many insects and arthropods feed on lichens, like mites, pseucids, and other groups. Snails are known to feed on lichens. So there's some studies.
But, many squirrels also. Yeah.
Mm-hmm.
Yeah.
So it is a new field of research actually where this type of work has not been done much, particularly in Northeast India.
Yeah.
Well, thank you, madam.
Thank you for your opinion.
Yeah, we'll go on to next question. Raja Prabhu, if you are there, you can directly ask your question to Dr. Robert. Raja Prabhu. Yeah, Rajappa, please. Unmute your mic. Will mute.
Good morning to one and all. So this is the nice presentation by Robert Lucky. You are one of my inspiration like analogy field. Sir, actually the how to identify or differentiate the residual lichens and ascolichens which is grow in tropical or subtropical area, because I am from part of southern Western Ghats from India. So we have seen many fungi or mushroom like appearance of fungus. So I can't conclude whether it is lichen or else in the fungi. Even I have carried some of samples, and still it is not recognizable whether it comes under the ASCO group or so. Clarify it, sir.
Okay, so I mean, there's two situations. If you have the actual spore-bearing structures, so the fruiting bodies, and it's obviously a lichen, then it's easy because you look at the fruiting bodies, if it's a basidiocarp or an ascocarp. So usually basidiocarps, there's only a few types of basidiocarps that are known in lichens. It's the normal, like mushroom type, for example, Lichenomphalia. Then you have these clavarioid types, like in Multiclavula and Sulzbacteromyces. And at first glance, they look like normal mushrooms, but you see at the base that they produce a thallus. And then you have Dictyonema group that have these resupinate corticoid hymenophores. So if they have these fruiting structures, it's very easy to tell if they are basidiolichens or not. If they are sterile, it's much more complicated because then you can only go by experience. So for example, if you have something that is filamentous and has cyanobacteria, it's most likely a Dictyonema, basidiolichen, because this is very rare in ascolichens. And I think there's very few species, very rare species known that do that in ascolichens. But if you have something like a green crust on soil, this might be anything. Yeah, so sometimes by the anatomy you can tell it could be a multiclavula or something. But if there's no fruiting bodies, it's very difficult to be sure if you have an ascolichen or basidiolichen.
Okay, thank you, sir. Thank you so much. And I have another one question too. So even in the morphological and anatomical study, we can identify the lichens in the recent taxonomies. But the current situation is expecting for only the molecular sequence taxonomic research. So here, is there any possibilities comparing the fresh samples and the herbarium samples to identify extracting the DNA and conclude whether species comes under this genus, this genera. So it is possibly to identify the species level, sir.
Well, I'm not sure if I understood the question completely, but I think the question went into difficulties to identify by morphology or phenotype, and then often you have to do molecular analysis and fresh material versus herbarium. So I think if you establishing like a taxonomic concept for a given area, it's very good if you can incorporate molecular studies. So you get a good picture of what you have in the area. But once you have your molecular data and you can incorporate the morphology of the sequenced specimens, you can establish a morphological taxon concept, and you can make keys based only on phenotype, morphology, anatomy for a given area. Because the most difficult challenge is if you use identification materials that do not apply to the area. For example, now it's very popular that people use proto-lichens of North America to identify lichens from India or from China or other areas. And of course, you will go wrong. I mean, you get possibly the right genus, but you don't get the right species. So if you do molecular studies with fresh material, you can actually confirm or check what species you have. And then once you have this, you can look at the morphology and then go back in herbarium and look at your herbarium collections and identify these based on the morphology. So you don't necessarily have to sequence herbarium collections if they're old. You can do this. Of course, now there's new techniques that you can use, but it's a very high It's costly and it takes a long time. And I don't think it's really necessary, but it's good to have a good concept that incorporates molecular data when you do like fresh inventories based on fresh collections.
Okay, thank you, sir. Thank you so much for that.
Okay, thank you. There is one question from Jagdish Ram. Using UV lamp in field for taxonomic recognitions, will it not harm biodiversity dependent on those lichens?
No, because I mean this is actually lichens producing these chemical substances is like a sunscreen. So usually in a normal light you have always a component of high energy UV light that could damage the photosynthetic apparatus. So these lichens produce these cortical substances that might be pigments. So the lichens are orange, yellowish, reddish, or brown, depending on the pigment types, or they produce these substances that have no color, but they can convert high energy UV light into visible light. So for example, if you, for example, simulate this by having a UV lamp and shining it on the lichen, what the lichen does, the substance does, it absorbs this UV light that could be damaging to the photosynthetic apparatus, but it converts it in visible light that is of lower energy and doesn't do any damage. So you don't damage the lichen by doing this. I mean, in any case, this is low energy, low level radiation that you have in the UV light. Of course, you shouldn't shine it in your eyes. That's not recommended, but it doesn't damage the lichens. Also, because you do it only a short time.
Okay. Yeah. One question from Imtilila Ching. She or he asking, any other bacteria, photosynthetic bacteria other than cyanobacteria in association with lichens as an algal partner?
No, it's not currently. I mean, there's only a few other bacteria that do photosynthesis, and these are very rare groups, and they actually have other types of pigments. They're more greenish than bluish green, and so far there is no lichen known to associate with these. So all cyanobacteria found in lichens, or all photosynthetic bacteria in lichens, are so far identified as cyanobacteria.
Okay. So,
Dr. Sanjeeva,
yes.
After you complete the question session, I will have some announcement. Okay.
Yeah. So Sebastian is asking, a compendium of micro and macro lichens key by Dr. Avasti is stated. The key from photobind may cyanobacteria or algae. It's difficult to go through for the identification. Is there any book for key or any method to find the cyanobacteria of algal partners? Some complicated questions. Sebastian, you directly ask me. After some time, I'll answer this question. Okay. So I want to clarify what exactly. The GP Sinha sir wanted to ask something. There is a report of yaks eating asnead lichens in higher regions of the Arunachal Pradesh. Okay, this is not a question. It's a response to
just I was telling because Dr. Jayashree has told. So there is first-hand information about the use of lichens. These yaks are grazing lichens, particularly when there is nothing. When the area is snow-clad, they are using profusely these lichens for their as a forage. That's all. Mm-hmm.
Yeah, actually, sir, this also I have come across also from Arunachal. Actually, this I meant earlier. The question it is mainly related to the Kaziranga National Park, where I have visited and saw lot of fructose lichen is used by bird and something. So this actually I was very inquisitive. So thank you for that information, and I recollected also this yak also eating asnia. Thank you so much. Okay.
Yeah, Dr. Sinha, you have any question to Robert?
No, it's fantastic. Just I was waiting for Dr. Robert's address, and its presentation is quite nice, and it is quite helpful for the youngsters. Thank you, Dr. Lukin.
Yeah, I have one question for Robert. You can say it's a final question. You started with folliculous lichens, then you took up raphidiceous lichens, then you're working. Sometimes you also worked on Parinocarpus. Now you're working on these basidial lichens. What's your future plan?
Can you repeat the last part?
You are now working on basidial
Yeah.
lichens.
The last question, what was it? Oh, the future plans. Well, whatever comes along. I mean, it's you cannot really plan. I mean, most of these things that have been working, they happened tropical ecology for a year, and then my professor told me I have to work on lichens. And why not work on folliculous lichens? I had no idea at the time what these things were, so I decided, okay, let's do this. And then other groups like the Graphidaceae and things, they came up just because, association with tropical lichenology, and these are very abundant groups. And then also, again, this basidial lichen story came up also out of chance because we were curious about in these Cora lichens and Dictyonema, because they said we have these different photobionts. They are filamentous or they are globular, and they're usually identified as different cyanobacteria genera. And so we wanted to find out if this is actually really two different genera or if it's just the microbiont that produces this different morphology. And so we started sequencing the photobiont and then also the microbiont, and then we found something is wrong with the microbiont. It doesn't really make sense. And we looked into this, and then this whole story with the basidial lichens popped up. And so it's really always predict. I mean, my experience is always when you go to meetings, when you meet other people and, discuss ideas, you brainstorm, suddenly something comes up and then a new project emerges. So I think these interactions with other people is very important.
Yeah, it's important to make people discuss with them, and also new ideas keeps coming. So I think there are no more questions, and there is one question in continuation of that bacteria. Till now, any reports on the anoxygenic phototrophic bacteria identified as an algal partner in lichens?
No.
The last question in chat box.
It would be difficult also. I mean, there's some ideas of proto-lichens, that lived very back in the past, hundreds of million years ago, that might have had very specific ecologies, but in excellent lichens, this is not known.
Okay, I think there are no more questions. We can wind up. Thank you very much, Dr. Looking, for your nice talk and interaction with Indian lichen and the participants. It's nice
Thanks
hearing.
for the invitation.
Yeah, and we Indians expect your cooperation, help, as you used to do for us in future also. Thank you very much.
Okay.
Yeah, actually, I'm just, it is an important announcement, Dr. Sanjeeva.
Please go ahead.
It is an important announcement. After this talk is over, and there is a oral and poster presentation. And after that, the 5 o'clock, we are having a validatory session. So I request everyone, the participants, the faculties, the scientists, and everyone present over here to attend this validatory session. And I request Dr. Robert also to join with us in this validatory session. And that session will be at 5 p.m. Okay. So thank you all. This is an announcement. I just requested everyone to be present. Thank you.
Okay, thank you everyone. So may I request Tanmay Sinha to mute Tanmay Sinha, mute your mic, please. It is disturbing a lot. Yeah. So now we will move on to the next lecture, next talk by Helena Fernandez. Okay. So let me introduce Dr. Helena Fernandez. She is an associate professor in plant physiology at the University of Oviedo, Spain. and her research focus on study of seedless plants, fern in particular. She received a PhD in biology in 1993 from Oviedo University, and she has also been researching fellow from the Roman Y. Kajal National Excellence Research Program, and she has been guest researcher at the Institute of Plant Biology, University of Zurich, and in many other places also. She is a member of Spanish Society of Plant Physiology and Spanish Society of In Vitro Plant Cells and Tissues, and she publishes regularly in relevant journals, having edited and contributed to a number of the study collections with Springer, and particularly I want to mention she is not Madonna. Okay, I tell you why, because when I searched for her photograph in the internet, that's what usually we do. Like, instead of troubling that person, we'll first see in the whether if something is available in the internet. So now when when her photograph was not available, I asked her a photograph. I said that in the internet it's not available. She specifically mentions I'm not a Madonna. Anyway, thank you very much, Dr. Helena, for accepting our invitation. And she has sent her recorded talk. I will play that here. Once the talk is completed, and you people can interact with her readily, and she will be happy to answer. And the title of her talk is that the gametophyte of ferns, less known phase of life cycle. So I'll play her recorded talk.
Bonnie, it's a great pleasure and great honor
to take part in this webinar, which I hope to be very fruitful
for the audience. I confess, I very much love your country. I wish to be there and not here, so distant. Well, I do hope to visit it sometime in the next future. Why not? In the next minute, I'm going to talk about the gametophyte of ferns, which I think it is the less known phase of the first life cycle. So, my main goal will be to show you the interest the gametophyte of ferns might have to helping deciphering some cues on plant reproduction. First of all, I would like to thank Professor Pratat Singh for the kind invitation. Professor Pratat Singh was very, very nice to cooperate with me in a book on ferns. I'm very grateful for that. Also, I would like to thank to Dr. Nayaka for his kind and encouraging support I received from him all these days. As you are going very soon to realize, my English is not as fluent as I would like, unfortunately. I wish the writing in the presentation could help to follow it. Perhaps I have to have rejected the invitation, but I love so much ferns that I confess I was unable finally to say no. Anyway, apologize for it. I'm going to rescue here what I have written in the abstract, which is more or less a declaration of intention of the real purpose of this intervention. Extant ferns represent a valuable genetic legacy, as they are among the oldest group of plants on Earth, being descendants of the first plants that evolved vascular tissue around 410 million years ago. Most part of published information so far focuses on the existing biodiversity in ferns, referring to aspects such as their phylogenetic relationship, taxonomy, morphology, ecology, or evolution. However, we are recently seeing a continuous drip of works that account for the contribution these plants can have to deciphering the cues operating behind plant development, and also to take benefit from their biotechnological application in different fields, such as the ornamental industry, agronomy, environment, medicine, etc. The purpose of this talk is to show up all the possibilities this plant group offer to illustrate the current research coping with this plant group, focusing on the less known generation, which is the gametophyte. To start with, I'm going now to remind all of you some features of ferns. Ferns are the second largest group of vascular plants on Earth, with almost around 11,000 species. Well, this number changed depending of the authors. They originated more than 400 million years ago and dominated the Earth's surface until 260 million years ago, which were overtaken by gymnosperms and angiosperms. They grow by everywhere, in forests, rainforests, and oceanic islands. They survive atmospheric conditions with more CO2 than they do now. First can cope with difficulties such as drought, salinity, and soil contamination with heavy metals. They are not usually attacked by insects. They contain a great arsenal of secondary metabolites like flavonoids, alkaloids, or phenols, with several properties such as to be antibacterial, anti-diabetic, anti-cancer, antioxidant activity, etc. Ferns have no flowers and no seeds. As you can see in the picture, the gametophyte lives separately from the sporophyte, and it is committed to carry out the reproduction process either by sexual or asexual means. Fecundation in ferns is really very primitive and needs water to allow Antheridia swimming until arriving into the archegonia, when is the egg cell. However, the life cycle of ferns have some variation. On the one hand, the gametophyte can arise a new sporophyte without the intervention of gametes, which is named apogamy, being compulsory in up to 10% of fern species. The ploidy level doesn't change, and gametophyte and sporophyte have the same. On the other hand, apospory allows to form gametophyte tissue from sporophyte directly without any sporogenesis process in the middle. This way, we can get gametophytes with increased ploidy levels. Well, I would like now to highlight the enormous interest for hemp. The more and more these plants are deemed as suitable models to depend on plant development. At this regard, ferns are ideal to study how land plants can maintain separate developmental programs that produce so dissimilar ontogenies and morphologies. Likewise, the evolution of development abbreviated as Devo, has opened a very interesting field in the last years. To understand plant development can be achieved by using a short number of model species. It needs also to pay attention to all the most representative plant groups. So, I rescue here this paper from Dr. Blackett et al., which point out the need to find all those missed links in the evolution of shoots between bryophyte and angiosperm, for instance. As you well know, our mother birds are descendants of the pioneer vascular plants. No doubt were here before dinosaurs. The global warming is a big concern for humankind, demanding to be solved. At this regard, I had the opportunity to meet Dr. Jaume Flexas, who is interested in exploring possible secrets hidden in ferns which could help to cope with this question. According to this author, ferns arose when the levels of CO2 were high, and then they never stopped to diversify. Given the increase in CO2 we face today, it might be expected they could respond better than the more modern groups of plants. Despite the interest aroused by ferns, there is a great concern we have to highlight, which is the lack of genomic data due to the complex these genomes are. Sequencing the genome first could provide valuable information to advance the knowledge of the functioning of plants. Sometimes researchers have to appeal in crowdfunding to go ahead with the goal. That's the case of Doctors Fywell and Pryor, and the case of Azolla, the fern Azolla. Let's now look at the application the study of ferns could bring to us. Next, I will comment at what point they are important for our routine life. To start with, biofertilization is a critical step in agriculture. In concrete, the fern Azolla has important repercussions within science of deep research in the last years. The symbiosis between the fern Azolla and the cyanobacter and Avaena has even higher nitrogen fixation than that of legume plus rhizobium. So there is a great interest to deciphering this symbiotic process. Another important question is to fighting against pathogens, which is attacks plants must face daily. Ferns and mosses are rarely infested by phytophagous insects in the wild. Insect infestation on ferns is 30-fold lower than in flowering plants. This tolerance is being used to create GMOs, as in the example of cotton, in which a protein, the protein TMA12, obtained from the species Dictaria macrodonta, was transferred to cotton to fight against the whitefly. Also, phytoremediation could take benefit from ferns. The species Pteris vittata is a pioneer in the research area, as it is a hyperaccumulator of heavy metals such as arsenic. Understanding the genetic and cellular mechanisms that allow arsenic and other heavy metals to accumulate and tolerate means being able to implement it in plants of interest. Perhaps nanotechnology results very attractive to me. The role of first in this new very important field. As you well know, nanotechnology promotes the using of very small particles to improving interesting properties of materials, such as their surface, solubility, etc. These nanoparticles can be made by different ways. For instance, mixing some extract with salts of metals like silver or gold. Amazingly, nanoparticles can be made from fair biomass as spores, and this way, any bioactive compounds they have could be much more effective. It's widely known that furs are a great source of drugs for using in traditional medicine especially, but also currently. The great arsenal of secondary metabolites includes many substances having therapeutic potential, like to be anti-cancer. Other times, as I will mention next, they can also be carcinogenic. Since long, furs are mostly appreciated by their beauty, contributing enormously to the trade of ornamental plants in many countries all around the world. In this sense, Nephrolepis was one of the most propagated plants, with one of the highest production rates. I would like to mention here also a very interesting input received from Professor Pratak Singh in a book devoted to furs I have edited in 2018. Finally, I should mention that furs are demanded also as food, being included in the diet of many people in the Asian and American continent mostly. However, as far as I know, I recognize it is not the case of my country, Spain. They are used to make fur salad, for instance. Some species are really very appreciated. Apart from the benefits, sometimes there are physiological characteristics such as large fronts, persistent rhizome, secondary metabolism, etc., make them tough competitors. This is the case of Pteridium aquilinum, named bracken, the 5 vascular plant most widely distributed all around the world for the last 23.8 million years. It belonged to open forest community long before human impact on forest and landscape took place. However, today, the prevalence of bracken has expanded considerably, partly due to human land use, but also due to the natural aggressiveness of the fir towards competing grasses, herbs, and trees. In addition, bracken is toxic to livestock and humans. The overall geographical distribution and the local abundance of bracken seems to be increasing in many places of the world due to several causes like cultural modifications. Climate change seems to favor bracken spread somewhere, and given the negative consequence this fur might cause to humans and animals, care must be taken to avoid overexposition to its dangerous chemicals. Bracken might cause gastric cancer and other types in cows, sheep, and horses, and even in us, due to the presence of tachyloside. There is a great concern, however, in my country, there is an important lack of information and good management of this fur. Here in the picture, it is shown the successive reactions from the tachyloside until forming the very dangerous DNA adduct, which affect seriously the genomic machinery. One revises the transcendental role furs might have for other organisms and also the environment. Let me go now to focus on the gametophyte of the experimental system. In other words, what can this unknown part of the first life cycle offer us? The gametophyte could contribute to a better understanding of alternation of generation, as it was aforementioned, and also to processes linked to reproduction such as gametogenesis, fecundation, and embryogenesis. Unfortunately, and given the complex genomes they have, first are still poorly sequenced, which is a great impediment to go ahead. The gametophyte at first is in the middle of the complexity existing between bryophytes and angiosperms. Gametophyte development goes from the one-dimensional stage or filament to the two-dimensionality, firstly as a spatula until reaching the mature hair shape. When mature, the gametophyte is a cellular monolayer, which can be thicker in the middle zone, showing an apical basal polarity and a dorsal ventral asymmetry. Organogenesis involves the formation of a meristem in the apical notch, rhizoids at the basal part, the sexual gametangia, archegonia usually placed in the middle, and antheridia mainly on the bottom. In some species might be asexual propagules, such as gemma and branching. It's really easy to culture in vitro the gametophyte at first. Usually, we start from spores to avoiding contamination and confess that starting from a spore represents a guarantee of success. However, I would like to remark and insist here that to cleaning up the spores, we use sodium hypochlorite and never mercury. As you well know, this is a very dangerous agent, so there is no need to think of it. It is also good to add in some drops of a tensioactive compound, like, for instance, twin 20. The process of asepsis is to washing spores in a concentration of the sodium hypochlorite between 5–10% for 10 minutes, and then removing the remaining, washing with water until up to 3 times for 10 minutes each. I proceed with the solution change by using centrifugation and 2500 RPMs. To speed up the elimination, I recommend you the using of liquid medium, but you can also use semi-solid medium. The gametophyte at first has a great regeneration capacity. In the first photo on the left, you can see the homogenization of gametophyte with a Waring blender or similar to break up the gametophyte tissue in small pieces. At the beginning of the culture, and for 2–3 weeks, the culture becomes browning, and you can think that all is dead inside the glass containers. But soon, some cells will start recovering the division capacity to proliferate actively, and we can this way multiplicate our gametophyte culture. This treatment goes better in liquid than in semi-solid medium, and this method avoids to start the gametophyte culture from a spore all the time. Well, in the next slides, I'm going to talk about my own research with the gametophyte, which is to study the molecular basis of apogamy in the gametophyte of Bryopteris affinis subspecies affinis. The question actually was from where we should start. Well, apogamy is a peculiar case of apomixis. And apomixis is a reproductive type where seed production occurs without the fusion of gametes, resulting for generations in offspring that genetically is a faithful copy of the maternal parent. I rescued this paper from Professor Wally Klaus, with whom I wrote this research, and in which it is considered how apomixis could allow the transgenerational fixation of phenotypes in hybrid plants. Well, as you can see in this picture, apomixis in seed plants, better than agar plants, this term, can proceed by the following processes. Firstly, we have the adventitious embryony, when cells older than megaspore mother cells form spontaneously an embryos inside the ovary. Then we have the diplospory, when megaspore mother cell abort meiosis before forming the embryo sac, whose egg cell becomes unreduced. Apospory is when cells from the ovary tissues form an embryo sac whose egg cell becomes unreduced. And finally, apogamy is when cells other than egg cell, antipoids, synergies, etc., inside the embryo sac will form an embryo. I would like to let clear here that apogamy in angiosperms is not a natural process as in ferns, but induced in the lab, which is known as androgenesis and genogenesis, from the male and female gametophytes respectively. There are many apomictic species with high economic value, but they are not all, of course. Researchers from several labs in the world are willing to control apomixis in those sexual species having an enormous economic interest. An amazing thing is that apogamy is very frequent in ferns, more than in angiosperms. And for this reason, I think the apogamy process in ferns deserves to be studied. In concrete, in ferns, apomixis combines a somehow case of diplospory, by which it is formed and reduced spores, and apogamy, by which an asexual embryo is derived from gametophytic somatic cells. After 1-2 months from spore culture, it is evident the organization of a brown center in the apical notch of the gametophyte, remarked by the red circle, which is an apogamous embryo, forming a new sporophyte with the same ploidy level than the gametophyte. In our lab, we have worked a lot to check the effect of phytohormones on both vegetative and reproductive gametophyte development by using several compounds of the main families. In concrete, in this experiment, we tested some valence auxin cytokinin by including the auxins naphthalene acetic acid and indole butyric acid, the gibberellins GA3, and also the polyamine spermidine. Apart from them, we have also tested some inhibitors of their biosynthesis or transport, like the naphthylthalamic acid, fluorprimidol, and cyclohexylamine. Naphthylthalamic acid is a transport inhibitor of auxins. Fluorprimidol is an inhibitor of gibberellins biosynthesis, and cyclohexylamine is an inhibitor of polyamine biosynthesis. In this slide, it is pointed out the effect of all of them on the percentage of two-dimensional gametophytes on the left, and also the length-width rate of gametophyte on the right after 2 months of spore culture. Here, in this slide, it is showing the effect of these compounds on the percentage of apogamy and on the embryo sites. Phytohormones affect both the vegetative and the reproductive development of these apogamous gametophytes. In this slide, we summarized some of the consequences caused by them. We can say that auxins like naphthalene acetic acid with cytokinins promote two-dimensionality, while apogamy increased when adding the auxin indolbutyric acid to the culture medium. Auxins seem to be relevant also for the placement of rhizoid on the basal part of gametophytes. Gibberellis are important too, promoting elongation. In this culture took place also the formation of callus tissue, which have proliferated actively by adding the auxins 2,4-D with kinetin. You can see on top in the right image the presence of green lipid bodies stained with Nile red. In presence of the auxin indolbutyric acid with cytokinin, the callus proliferate too, but the aspect is quite different. Green, more morphogenic. We are going to use this system in the next future to deepen on this morphogenic capacity. Apart from it, it is always very useful to have a good protocol for inducing and proliferation callus, and also that callus have regeneration capacity. I mean callus deals with organogenesis or embryogenesis so that we can face finally transgenesis experiments. Well, apart from the physiological study, we drove as a formed unit a molecular approach to the apogamy event in collaboration with Professor Willy Gross-Niklaus and Jonas Grossman from the University of Zurich and the Functional Genomics Center in the same city, respectively. Omics technologies based on comprehensive biochemical and molecular characterization of an organism, tissue, or cell type, and next-generation omics approaches facilitate the analysis of non-model organisms owing to the rapid generation of large amounts of de novo systems biology data, making them attractive options for studying plant development and evolution. To start with, we had to face the culture and collection of gametophyte samples. For this purpose, we considered one kind of sample that composed of filaments or one-dimensional gametophytes, and other sample including the spatula and hair shapes of two-dimensional gametophytes. The first one were obtained from crowded culture of spore in liquid media, and the second one were obtained upon being transferred the filament to semi-solid MS medium, letting them quite a bit more time in culture until ready. The culture of gametophytes are quite asynchronous. That means we had to check the developmental stage and pick them up under the microscope. Thanks to the support provided by Dr. Jonas Grossman, who is expertise in bioinformatics, we could publish the first transcriptome done with an apogamous first, Diopteris affinis subspecies affinis. In fact, it was the result of joining proteomic and RNA sequence data, which is called proteogenomics, increasing up to threefold the number of identified proteins than when only we match against the Viridi database. The most part of the matches came from Arabidopsis. We identified 1,397 protein clusters. As you can see, we detected proteins involved on reproduction related to organs first don't have a cyst, and also proteins associated to gene silencing, for instance. The results obtained deserve to be published in this journal, as you can see. In this paper appears all the methodology used in case anyone could be interested. One step further was to compare the proteomic profiles of 1 and 2-dimensional gametophytes. I would like to let clear that the transcriptome was done joining either filamentous, spatula, hair-shaped gametophytes. In this second study, the samples were analyzed separately to compare the profiles in the 1 and 2-dimensional gametophytes. All this was committed to detect change as apogamy is present in the 2-dimensional gametophytes but not in the filaments. The results revealed that proteins upregulated in 2-dimensional gametophytes are implicated in the regulation of meristem growth, auxin signaling, reproduction, and sucrose metabolism, among others. Likewise, proteins upregulated in 1-dimensional gametophyte are enriched in stimulus and defense genes, as well as genes involved in epigenetic gene regulation and ubiquitin degradation. Let me now mention our last proteomic analysis, which were done to compare the proteomic and physiological profiles in 2 related ferns, the Apogamus Dryopteris affinis and its sexual progenitor Dryopteris oreades. By using the software string, the proteins were then analyzed according to the GO ontology in the 3 categories: biological process, molecular function, and cellular components. Apart from all this, string is a very useful tool to providing the interaction network existing among proteins. Here, it showed the enrichment of proteins according to the 3 categories: biological function, molecular function, and cellular components, corresponding to its reproduction models. The proteomic analysis counted 879 proteins, and after annotation, different regulation was found in 206 proteins of the Apogamus species and 166 of its sexual counterpart. The proteins upregulated in Dryopteris affinis, the Apogamus species, are mostly associated to protein metabolism, including folding, transport, and proteolysis, ribosome biogenesis, gene expression, and translation, while in the sexual counterpart they account purely for starch and sucrose metabolism, generation of energy, and photosynthesis. Apart from the proteomic analysis, we quantified by using ultra-performance liquid chromatography coupled to mass spectrometry, flash generation, up to 14 phytohormones, among them auxin, cytokinin, gibberellins, abscisic acid, salicylic acid, and brasinosteroids, to compare their profiles, on the one hand, in filamentous spatula hair-shaped gametophytes of the Apogamus species, and on the other hand, we compare also their content between the hair-shaped gametophyte of both species, sexual and asexual. The results found that the auxin indolacetic acid, the cytokinin transceatin, transceatin riboside, isopentenyl riboside, the gibberellin GF4, the brasinosteroid castasterone, and the abscisic acid accumulated more in the sexual gametophyte than in the Apogamus 1. Then, when comparing the 3 Apogamus states, the cytokinin, benzyladenine, and salicylic acid peaked in filamentous, the gibberellin GF3 and brasinolid in the spatules, and the hydroxyatine riboside in the hair-shaped gametophytes. Of PCAB plot, principal component analysis, as you will see, you can better appreciate how the most part of variation up to 90%, group it splitting sexual and asexual gametophytes. And then, on the sexual, also among the three developmental stages, filaments, spatula, and hair shapes. The black line is to help you to remark the zero. Currently, it is necessary to find alternatives to the sources of raw materials and energy that we use because they can run out. First are little studied plants but with interesting characteristics. So, for this reason, we decided paying attention besides to those proteins found in common in both species of the previous experiment. This is a recent study done by a master student. From the total of proteins, there were 417 equally present in both species of ferns. These proteins were linked to biological processes such as response to stimulus, being mostly component of chloroplast and nucleus and whose molecular function was mainly enzymatic or binding. Apart from relevant proteins involved in plant development processes such as reproduction, response to phytohormones, or gene expression, we would like to remark those associated to respond to stimulus or defense, which could have important biotechnological repercussions. The results obtained are included in a new publication, which I hope to be sent soon. This kind of research could represent to achieve the transgenesis to plants of genes of agronomic interest involved in tolerance to abiotic factors such as cold and heat, drought and hypoxia, salinity, heavy metals, or ultraviolet radiation. In addition, they can be also useful to fight against pathogens. Likewise, genes associated to metabolic pathways linked to pigments production, for instance, could benefit the ornamental industry of flowers. Please, with your permission, I would like to share with you these books published since 2011 by Springer, in which we try to compile all the last findings in this plant group in several fields: biology, biotechnology, evolution, biodiversity, conservation, genetic and metabolic resources, and also environment. I'm delighted to show you the co-editors and also your neighbors who have made possible the last book, our last book, to be published on next November. Please let me now to mention people helping the research done with FIRST along the years. I would never have been able to do anything without them. Therefore, I must express my gratitude for the collaboration provided by Professor Welly Brownie-Klaus, Dr. Valeria Gagliardini, and Dr. Stefan Weider at the University of Zurich for having introduced me with the omics, as well as to Dr. Jonas Grossman of the Functional Genomics Center of Zurich for being so helpful with the bioinformatics issues. My thanks to Drs. Isabel Feito and Lucia Rodriguez from the Cerida Center here in Spain for helping with the phytohormones analysis, and finally to my colleague at the University of Oviedo, Professor María Jesús Cañal and Dr. Alejandro Rivera. Well, finally, many thanks for your attention, and please don't hesitate to contact me if you need to clarify anything, and I promise to be better writing than speaking in English. Many thanks.
Yeah, thank you very much, Helena, for a nice presentation. Now it is open for discussion. If you have any questions, you can directly ask to Helena, or you can type in the comment box. Let me check in the comment box if there are any. No questions in the comment box. Any questions for Dr. Helena? Is that Dr. Benjamin? Benjamin. It seems there are no questions, Dr. Helena. So I thought there would be some question, at least in the question, this comment box. So, okay, Dr. Sachin Patil is asking, is there any synthetic media specific for germinating the teridophytic spore?
Yes. Hello, everybody. Yes, but no specific. In fact, to face the spore culture, to start spore culture, it is very useful. Just MAs, the Murasigan school media, widely recognized and widely used all around the world in this when we committed anything in vitro culture. So is a very good media. There are also another solutions like the knob medium. I think you will know, very sample, and others, but in general, full strength or half strength MMS media is good enough to culture sport. It's really very easy.
Okay, thank you. Dr. Divakar, you have something to say? Dr. Divakar is also at Spain. He works at University at Complutense.
Hi, Sanjeev, can you hear me?
Yeah, I can hear you.
Hi.
Sorry, what is it? Hello?
Hi, Elena.
Hi. Good morning here.
I'm good.
Yeah. Yes.
Really, first of all, I thank you very much for accepting the invitation, Sanjeev, and joining this session and sharing your knowledge to the teridologists and all the scientists working in India. It's really our great pleasure to have you in the room today. Yeah.
Many thanks.
A very nice talk, Elena. Very nice talk. Yeah.
Thanks. Perhaps a bit surprising. I think the first are not very well known. I think, but it's hopefulness. Very kind.
Yeah, maybe. There's one more question from Sachin Patil. Madam, can you share your email address? Okay, email address is there. It is already printed in the abstract book. So once you get the abstract book, Helena's email will be available to you all.
But my email, I put my email addresses in the last picture. Anyway, if you want, I can write here. This is Fernandez Elena. Sorry.
Yeah, go ahead.
Sorry. Doc is. Sorry. It's not correct. Sorry.
Wait. Let me show it. Yeah, this
now it's correct. Yes.
This is the one. Okay.
So don't hesitate to write me, and I.
In any case, thank you very much, Dr. Helena, for sparing and for your nice presentations. And
It was a pleasure. It was a pleasure. Sorry for my English is not good enough, but well, more or less, I think I could explain the last research done with first a little bit at least.
yeah, even
That's
Indian English is also not that good, and it's okay. But what is important is that it should be whatever you say should be understandable. That's it. So,
Okay.
Dr. Sanjiva,
yes, ma'am, please.
yeah, I request Dr. Helena to be
Hello?
there in the validatory session.
Oh yes, yes,
Yeah,
yes.
yeah. Yeah, I request you to be present there. Okay, yeah.
Okay.
Okay.
Thank you. We'll close it. We will break for lunch. Quickly after the lunch by 2 o'clock, we are having the presentation from the participants. Sharp by 2 o'clock, everybody be ready.
Dr. Sanjiva, I told you regarding the chair and rapporteur, okay?
I have
Yesterday.
to, yes,
So
yes,
you
I have,
please, you will announce that. Yeah. Okay.
yeah. See, the chairman for the oral presentation is Dr. D.K. Upreti, and he will be helped by Dr. G.K. Mishra. And for the poster presentation, chairman is Dr. J.P. Sinha, and he will be helped by Dr. Silchar Joseph. So we'll meet in the afternoon after lunch, sharp by 2 o'clock.